right, so thank you so much for the introduction, and thank you all so much for being here. I'm thrilled to talk to you today about how some new image analysis tools in the field of molecular neuroimaging can help unveil the role of serotonin in depression and suicidal behavior. So serotonin is a chemical in the brain that helps regulate a range of processes, including mood and cognitive functioning. It's often called the body's natural feel-good chemical. And I say natural because it's actually produced in the brainstem. And then neurons that connect the brainstem up with the rest of the brain will release serotonin all throughout the brain. I wanted to start by giving you a brief history of serotonin. So it was first discovered in 1948, serendipitously in the search for a cause for high blood pressure. This mystery chemical that they extracted from blood, they thought at the time it was a vasoconstrictor, so something that tightens the vessels. And so that's where the name comes from. So sero from serum, blood serum, and tonin related to vessel tone. And it wasn't until a few years later that they realized it actually was a neurotransmitter, so a chemical messenger in the brain. So the connection between depression and serotonin dates back to at least the 1960s, where there was a hypothesis that clinical depression could be caused by deficits in serotonin. And then this was bolstered by evidence finding lower serotonin in the brains of those that had died by suicide who had had clinical depression in their lifetimes. The first drugs that selectively regulated serotonin in the brain came about in about the 1970s, and then some of the first molecular brain imaging studies followed. The first selective serotonin drug that was FDA approved for depression came in 1987, which was fluoxetine. It's a selective serotonin reuptake inhibitor, or an SSRI, and it seeks to boost serotonin in the brain. And then right after that were the first published studies of molecular brain imaging with depression. So overall, the idea is that when serotonin is low in the brain, there might be more depression symptoms and potentially suicidal thinking and suicidal behavior. So though serotonergic drugs have been prescribed and have been out for decades, the current drugs that seek to increase serotonin in the brain only help about a third of people suffering from depression. And so, of course, we want to do better. So our team is working to find biomarkers, identify biomarkers that can help figure out who might respond to certain treatments and who might go on to develop depression or develop suicidal behavior. And the goal is to alleviate suffering from depression and suicidal thoughts and behavior before it occurs. And that's great. So I'm going to be presenting on measuring and quantifying serotonin in the brain with a tool called positron emission tomography, PET. So this is a PET scanner. Our research participants lie down in the scanner, and one of our team members will inject a radio tracer via IV into their arm. That radio tracer will travel throughout the body, first going to the heart, and then to the brain as well as other locations. And then from the energy emitted by that radio tracer in the brain, we can record images of the brain. And so this specific PET study I'm going to show first, we got 2 PET scans in each individual. And PET scans are really expensive and they're really complex, and so it really makes it a unique data set that we have these 2 different scans targeting different components of the serotonin system in the same person. So the first looked at density of serotonin transporters, and the second looked at serotonin 1A receptors. So when serotonin is low in the brain, it will drive the density of serotonin transporters down. When serotonin 1A receptor levels are high in the brain, it will drive serotonin down. So now I want to show you what these images look like. So these are average images over all depressed participants in the study. The left is the transporter, the right's 1A. I'm showing you one hemisphere where the top looks at the outer surface of the brain, and then the bottom looks at the midline. The red shows higher levels of each of the different receptors. And what was striking in this is that if you just look at this simple cartoon schematic of how serotonin is supposed to look in the brain, you can see in the center where it's more purple is where serotonin is actually synthesized within your brain and made in the brainstem. And then there are neurons and fiber tracts that project forward and then wrap around and towards the back of the brain. And so if you look at those bottom images from our actual participants, you can see those bright red areas, like right as the fibers project out and then as they wrap backwards. And so it was observations like this that really made us want to look at the brain from a serotonergic perspective. So a lot of the times in neuroimaging, we define brain regions, for example, by structural landmarks, so looking along these hills and valleys throughout the brain, or by more functional landmarks, like you may have heard of the hippocampus, which is called, some people call it the memory center of the brain. But we really wanted to look with specificity for the serotonin system. And so this brain atlas came out of Copenhagen that used data from hundreds of participants in their PET scans and give us these 10 brain regions that have distinct serotonergic functioning. So for example, the green region might have very high levels of one serotonin receptor and average levels of the rest, whereas the blue region has very low levels of all serotonin receptors. And the idea is that if we can quantify the level of the transporter or the 1A receptor in these specific regions that are tuned to the serotonin system, we might have more power to detect effects. So then on to the clinical part of it. So both Sarah and David gave amazing introductions about EMA. So I'll just very briefly touch on it. Smartphone surveys given multiple times a day for the course of a week, and we asked about the experience of these different daily life stressors since the last prompt. And then looking over the course of the week, we computed a summary metric for kind of daily burden of life stress. So combining the brain imaging with the clinical EMA, we come to the results. So for the serotonin transporter, we found effects in this focal area in the back of the brain that was specific to currently depressed individuals with a history of suicide attempts. So that's shown in the red-orange line, whereas no effect was present in those without a history of suicide attempt. So specifically in individuals that reported the highest levels of stress, they had the lowest levels of brain serotonin transporters, which is indicative of having low serotonin. And then with the second PET scan, we saw a much more global effect. So in 9 of the 10 regions, everything except that frontal area shown in gray, we saw, again, this effect that was specific to those that had a history of suicide attempt that was absent in those without. And here, in those that reported the highest levels of stress, they also had high 1A receptors, which is indicative of low serotonin. So summing this up, individuals with a history of suicide attempts with more daily stress had fewer serotonin transporters, more 1A receptors, but the serotonin system was not linked to stress in the non-attempters. And so taken together, we show low serotonin is associated with periods of highest stress in the attempters. And what makes these results really exciting is that if we can see that the serotonin is stress responsive, then we can potentially develop and administer timely interventions that can target the serotonin system and induce a resiliency to stress. And this can hopefully stop maybe the onset of depression or suicide attempts. So next, I want to delve deeper into the serotonin system anatomy. And so we're coming back here to these serotonin-specific fibers that are extending throughout the brain. And so I showed you this one image that kind of depicts this, and then in a separate set of individuals with different PET scans, I'm showing you a different way. So again, on the top, it's a midline slice, and here actually shows that brainstem area where serotonin is synthesized in the brain. And so you can see the white area shows the highest levels of serotonin transporters, so the highest levels of serotonin. And you can really see it kind of bleeding out into the front of the brain and swooping back. So our goal was to automatically delineate this serotonergic tract of fibers. And so we did that, which is shown in the blue. And then we parcellated up that blue tract into tiny segments so that we could quantify the level of serotonin transporters in each of the segments and look and see how the serotonin levels change as we move back through the brain in depressed and in healthy volunteers. Pressing the wrong button. Okay, so here are the results. So on the bottom, you can see that tract where it starts on the origin, where the raphe nuclei are in the brainstem, shown with the yellow star, and then you can see as you go right, it moving along that fiber tract. The vertical axis shows the serotonin transporter levels. Each individual line on this is an individual participant, and then the bold ones are the average of all the healthy volunteers and the average of all of the individuals with major depression. And so you can see that there's this deficit in the serotonin transporter closest to where serotonin is made, and the effects persist as you move through the frontal cortex, and it starts to dissipate as you move throughout that tract. And by looking at the serotonin system in kind of this non-conventional way, we were able to show an effect that was far greater than if you just look at individual regions on their own. So there was 27% lower serotonin transporter density in individuals with major depression relative to healthy volunteers. Okay, so the last analysis I want to show you is something I'm really excited about. It's a big data analysis, and it's something that we're working on right now. So this is all new data. So I've been talking about just general levels of serotonin transporters and 1A receptors, but with this big data analysis, we can get hundreds of different metrics of the serotonin system from each brain region. So I just put up a picture of a brain here, but on the right, this correlation matrix shows each of the different metrics we can get from one single brain region. And this just looks at how the metrics are related to each other. So some are really highly correlated in yellow, some are less correlated in red, but it gives us the ability to kind of detect distinct information from combining all of these different measures. So this tool is called radiomics. And it studies images by measuring patterns and shapes and looking how small parts of the images connect and relate to each other and how the signal changes. And then we can turn it into analyzable data. And what's really exciting about this is that it's already led to FDA-approved tools in oncology. So this specific tool is used for treatment planning. But it's rarely been applied in psychiatry. So we saw a real opportunity to combine this molecular brain imaging with this new big data technique to kind of harness more power. And so our goal was to use radiomics and combine it with machine learning, or AI, to build on past findings of high serotonin receptor binding found in those with depression. So coming to our results, with this radiomics analysis, just looking at one single region of the brain, we get 80% accuracy to distinguish major depression from healthy volunteers, which was far superior to that if we look at just the standard transporter level in the raphe nuclei. And then we also had higher sensitivity to detect who was depressed and higher specificity to detect who was not depressed than the raphe nuclei alone. And so we were really excited about this 80%, but we want 100%. And so we're working right now to optimize and enhance this tool to try to boost that signal. And so right now, with our current data, we're ready to use radiomics to predict depression severity, to detect who had a lifetime history of suicide attempts, and response to a specific antidepressant medication. But my dream is to use radiomics to predict who will develop depression before the actual onset and who will go on to make suicide attempts, so that we can really target personalized interventions to try to stop these things before they occur. So to sum up, molecular brain imaging has been used for decades in psychiatry. We still desperately need biomarkers that can help identify who needs help and guide how to help them. And so we're developing novel image analytic tools that we're hoping can provide critical insights for treatment so that we can intervene before depression and before suicide attempts happen. So I wanted to thank everyone so much and thank my mentors, and I'll take questions.